Hi there and welcome to today's video. Today you will learn about the early stage of synthetic biology and genetic engineering by having a look at bacterial transformation. But before we take a look on this method, what are the differences between synthetic biology and genetic engineering? Synthetic biology is an interdisciplinary area that involves the application of genetic engineering principles to biology. It aims at redesigning and fabricating biological components and systems that do not exist in the natural world or are altered in a desired way. In this process, chemical synthesis of DNA is combined with growing knowledge on genomics to enable quick construction of DNA sequences and assembly into new genomes. Genetic engineering involves the transfer of individual genes from one cell to another and therefore changing a naturally existing code. Synthetic biology envisions assembly of novel microbial genomes from a set of standardized genetic parts that are inserted into a microbe or cell, making something not naturally occurring. There are many methods to genetically engineer different kinds of cells. In this video, we will look at the most basic method, the transformation of bacteria. Just like more modern genome editing techniques, traditional bacterial transformation has been borrowed from bacteria. In nature, when one bacterial cell dies, its genetic material is released to the outer environment. Other bacteria are able to take up this genetic material and insert it into their own genome. This process is called transformation. It allows bacteria to acquire genetic information not only from parents, but also to take up new genes during lifetime. Not all bacteria are able to do this naturally, only the so-called naturally competent bacteria can. But scientists found a way to induce this ability in other bacteria. This is called induced competence. Inducing competency enabled scientists to add genetic information also to other bacterial strains. But before we can add genetic information, we need a vehicle to transport it into the cell. This vehicle is called a vector, and there are different options available. For example, phages, viruses that target bacteria and can be used to insert DNA into a bacterium. Most often, a circular, ring-like piece of DNA that contains the information we want to add is used. This is called a plasmid. To then transform our cell with a plasmid, we first make them competent. For this, the salts calcium chloride and magnesium chloride are added, which shields the charges of the DNA backbone. This enables the plasmid to curl up in a small ball, as well as to cover the charges of the bacteria wall so it doesn't repel the plasmid. Then the cell wall is made more permeable, so the small curled up plasmid has an easier time to slip in. This can be done, for example, by heat shocking or by adding electric pulses. However, this process is not very effective and only a small portion of bacteria will take in our plasmid. So how can we separate the ones that did from the ones that were lazy and didn't? This is done by using a selection marker. Oftentimes, antibiotic resistance is used for this step. In our plasmid, there is not only the gene that we want to have inside the cell, but also other genes. For example, one that gives the bacterium the ability to survive specific antibiotics. Now we can let the cells grow on a surface that contains nutrients and the specific antibiotics. Since only our transformed cells that use the plasmid can tolerate the antibiotics, only they are able to grow. That's how we know our transformation worked. Now that we know the bacteria contains our gene, it has the instructions from the blueprint to produce the protein for us. These days, many people benefit from the discovery of bacterial transformation. This principle is, for example, applied to make large amount of insulin, which can be used to treat people with diabetes. Although there's no cure for diabetes yet, this fantastic technology has provided a way for efficient insulin production that relieves their symptoms. Next time we will discuss about more recent genome editing techniques that may revolutionize the future of health. Don't forget to subscribe to our channel and follow us on social media to stay tuned.